Welcome everybody to this morning's JSPS Pathology Grand Round session. This is sponsored by the Shanks Foundation and the Pathological Society. Please can I remind audience members that following the lecture there will be a question and answer session. So please can you write your questions in the Q&A box at the bottom of your screen and I will read them out at the end of the lecture. So today it gives me great pleasure to welcome Professor Sir John Byrne. Sir John is a Professor of Clinical Genetics at the Translational and Clinical Research Institute and leads the Cancer Prevention Programme Research Group at Newcastle University. After obtaining his degrees, he became Professor of Clinical Genetics in 1991, and before that he was appointed as a consultant specialist in Newcastle in 1984 and led the regional NHS Genetics Service for 20 years. He helped to create the Millennium Landmark Centre for Life, and he's chair of a Newcastle-based DNA device company. He was knighted in 2010 and chosen as one of the first 20 local heroes to have a brass plaque on Newcastle Quayside in 2014. He's trustee of the European Hereditary Tumour Group, UNESCO NGO Global Variome and HUGO, the Human Genome Organisation based in London. He's currently Vice President of HUGO International. He leads the Cancer Prevention Programme Research Group at Newcastle University, and he was lead for the CAP2 and CAP3 randomised control trials. Today he will give us a lecture entitled Lynch Syndrome, Tumour Screening and Urogenital Surveillance. Thank you very much for coming to speak to us this morning, John, and over to you.
Thank you very much, Mark. It's an honour and pleasure to be with you. I'm just trying to share my video here, which says unable to start video. So I'll try again. Where am I? Okay. Can you see me yet or not?
We can see your lecture. It's not on full presenter view. It's on the view where we can see the slide, all of the slides on the left and the main slide on the right. I think, yeah, if you click on that, yes, that's fine. We can now see it all.
Excellent, thank you. And it's an honour and a
one more thing, John. We're not seeing your camera, so if you could click on your camera just to switch your camera on, I think that would be helpful as well, please.
Yes.
Yeah, that's it. We can see you now. That's perfect. Thank you, John.
Sorry about the stuttered start. Well, thank you all for having me to speak, and thank you, Mark, for that lovely introduction. I'm going to talk about Lynch syndrome, which is named after my old friend, the late Henry Lynch, shown here on the left. And Henry spent 20 years trying to get the world to realise that cancer could run in families, based on the family of Eldred Warthin on the right there, whose seamstress's family had a fairly dramatic history of endometrial, gastric and colorectal cancer. And basically, I've been involved in this now for 30 years. I'd like to share with you those first 30 years of research and then some exciting new developments, which I hope you will all be interested to be involved with. So these are my disclosures. Had Ted Naggs, a local GP, been a bit more outspoken, he might have had the syndrome named after him. He's seen here with members of our research team and members of the family which we called the Durham family. And that Durham family had, when he wrote it up, 45 cases of colon cancer in it, which was fairly dramatic. He wrote the paper with Dunstan and Naggs in 71, and it was largely disregarded by the genetics community, but we went back to try and map it, led by Tim Bishop in Leeds. And in fact, we found ourselves at the start of the maelstrom of discovering the genes underlying this condition. This is a paper from 1995 describing the MLH1 gene in this family, but in fact, we were already running with the other genes I'll come back to in a moment. I'd got into this because of familial adenomatous polyposis, a rare condition affecting about 1 in 8,000 people. Simon's typical example, presented to us as a teenager, and here at the age of 23, following his resection of his colon, with what's obviously a very large number of pre-malignant lesions. And we were helpful to Vicky Murday and Walter Bodmer and colleagues in finding the APC gene on chromosome 5. And in fact, we were using markers within the gene to track this family and tell Simon's sister that she was in the clear. And because of tracking down these families, we started to find these other families, which were clearly not polyposis, which we all call hereditary non-polyposis colon cancer. This was actually the other big family that we shared with Kolodner's group in Boston, the yeast geneticists who found MSH2. And Ted was a local shepherd who'd had multiple colorectal and skin cancers, and we originally called it Muir-Torre syndrome, which we now know to be a subtype of Lynch. And his sister had endometrial cancer. His granddaughter developed colorectal cancer at 18. And so we shared our DNA from Ted with the yeast geneticist. They pulled the MSH2 gene, found a mutation, which actually wasn't the pathogenic variant in the gene, but it allowed us to do what we think was the first diagnostic test using linkage within this family. We now track about 8,500 people with Lynch syndrome in the prospective Lynch syndrome database set up by Paul Muller from Norway. And we now know that about 80% of gene carriers will develop cancer by the age of 75. The exciting thing about the mismatch repair genes was that they were a well-studied group of genes which are used by all cellular organisms to do their DNA spell checking. And so as soon as we knew where the problem lay, we were very rapidly into being able to identify all of the genes involved. Interestingly, Stanley in the box there was our lead into this family. He had familial adenomatous polyposis and a learning disability due to a chromosome 5 deletion, which came from his father's side. His father, interestingly, had been on Christmas Island clearing up after the nuclear tests about the time he was conceived, which may or may not have played a part. But his mother gave him the gene for Lynch syndrome. And as a result, he got colon cancer 3 times before he was 30. This is my colleague, Chris Heinen from the University of Connecticut, who gave an excellent talk at a session I organized in America a few years ago. And he allowed me to take his slides and rapidly collect them together. So MSH2 joins up with MSH6 and spots any mismatches in the DNA. And then these clamps load onto the DNA and catch up with the polymerase. MLH1 joins with PMS2 typically to then activate those clamps. And then XO1 strips off the recently created strand of DNA until it gets back to the error, corrects it, and then runs the polymerase again. So basically, that's happening all the time in every cell in our bodies, and it's quite remarkable in a way that losing this gene doesn't cause more problems. Of course, you can get by on the spare copy, but the chances of getting a spelling error in that spare copy in your lifetime means the likelihood that you'll end up with cells which have lost both copies of one of these genes, and it's those cells that then accumulate errors in repetitive DNA. This is the core of the problem. This is the TGF-beta receptor 2 gene, a very important gene in cancer prevention. And it has within it what's called a microsatellite, an old term from the electrophoresis days, where you've got 10 A's in a row. And if you fail to correctly copy that and drop back to 9 and don't correct the error, then you will cause a frame shift, which means that all the amino acids thereafter will be different, and you typically come to a premature stop codon. And so if it goes back to 2, it goes back to 8 A's, then you end up with a different string of amino acids. And these are called frameshift peptides. And these are highly immunogenic because they're not recognized by the immune system. And indeed, Magnus von Knebel-Döberitz, my long-term colleague in Heidelberg, has been working with his colleagues on vaccine development for a long time. And there are now at least 3 different approaches to using these frameshift peptides as a trigger for vaccination. So the molecular pathways to colorectal cancer are ever more complex, but there are a couple of broadly important categories that are worth referring to here. First of all, FAP is the model for chromosome instability, losing the APC gene and then a string of other activating and oncogenic mutations. Conversely, microsatellite instability is the other common pathway, accounting for about 15% of all colorectal cancers. And on the bottom there, you can see the TGF-beta receptor 2 gene. About half of the ones that arise as a result of silencing of the MLH1 gene, which is the commonest cause, they will have the driver mutation BRAF V600E, which is useful in terms of deciding who you should do germline testing on. There's TGF-beta receptor 2, one of the genes that gets lost if your mismatch repair breaks down. So Lynch syndrome, in a way, is a genetic model for about 1 in 6 of colorectal cancers which involve breakdown of MLH1. Most of those, of course, are an acquired silencing, a methylation of MLH1 promoter. And so what we're interested in is picking out the ones which are due to a germline defect, or more rarely, an epigenetic defect where you've lost MLH1 typically early in development and you have all of the features of Lynch syndrome, but it doesn't run in the family. BRAF itself is a very important gene, described about 20 years ago by Mike Stratton and colleagues, and this is in the EGFR receptor pathway, and there is a point mutation, V600E, which changes the valine to glutamic acid at that position. What that does is effectively leaves BRAF switched on. So if you pick up that mutation, then you're going to carry on getting cell division being driven. And crucially, EGFR receptor inhibitors will make no difference to that tumor because it will carry on regardless. I was visiting families with FAP, and the one in the picture, Jonathan, was a little boy of 12, and we just scoped and he had no polyps, but his mother had a panproctocolectomy for FAP. And being trained as a physician, I got to thinking, wouldn't it be nice if we could treat him medically rather than surgically? And that gave me the idea of doing a genetically targeted trial. We had more and more of these families turning up, and they were highly motivated, got lots of pathology, were already under surveillance, and so we shouldn't need too many to do some sort of intervention. In fact, they lived right next to the Monk Wearmouth pit, and I popped in and got the miners to give me a picture of one of their canaries, which of course were used in the mine to detect gas before the humans fell over, and then rushed out to have resuscitation. In a sense, these families I saw as being canaries for the rest of us, because we know that the majority of colorectal cancers have loss of this same gene in a sporadic sense. So we set up a trial with European funding. It was concerted action in Europe, so we called it concerted action polyposis prevention initially. And it started in 1993, just as the genes for Lynch syndrome were becoming available to us. We immediately realized that it was going to be difficult to track these adolescents for many years over many countries and give them interventions. And of course, the Lynch families were much more attractive. And so almost immediately, we started work to persuade colleagues around the world to do an intervention trial on Lynch syndrome. It took us 6 years or 5 years to get it set up, and we started recruiting CAP2 in 1999. The two interventions we used were aspirin-resistant starch. This is Gabriel Kuhn, a physician in Melbourne who did a case control study and said it seems like people who take aspirin-like drugs get less bowel cancer. And that triggered a cascade of observational and epidemiological studies, which are summarized in this very nice graph here, up to 2000. And everything apart from one to the left of the line, saying that people who take a lot of non-steroidal anti-inflammatories like aspirin, and particularly including aspirin, will get less bowel cancer. The one outlier was the Paganini Hill study based on very elderly Californians in care homes, where it seemed to have a reverse effect. So it looked like aspirin was an obvious thing to try because we could do a randomized controlled trial with it. And fast forward 30 years or so, this is our paper in the Lancet of 2020, which showed the planned 10-year follow-up of the just over 1,000 people that we recruited into CAP2. And the effect was fairly dramatic. We already knew in 2011 that the per protocol group was seeing benefit, but by now the intention to treat was also there, and we had a significant intention to treat, which was quite impressive. But crucially, the incidence rate ratio, which takes account of all tumors rather than just the first one, which is what you get from hazard ratio, shows a really quite dramatic effect of a 50% reduction in the number of colorectal cancers among people who took 2 aspirins a day for between 2 and 4 years. And that effect, remarkably, is still apparent at 15 to, in fact, heading into 2 decades. We only planned to follow them for 10 years, but in Britain and in Finland, we were able to use cancer registry data to add on the second half. What that means is that for every 25 people who took part and took 2 aspirins, we prevented 1 colorectal cancer. Crucially, there was no significant difference in side effects in this relatively young group of mean age of about 45. And off the back of that, we actually got permission, or NICE gave a recommendation that everyone with Lynch syndrome should be recommended to take aspirin, although at the moment only about a third of them do, because people are very anxious about the 600 milligram dose. It's actually a subanalgesic dose. It's not a big dose, and in young people causes minimal side effects, especially if you get rid of blood pressure and H. pylori. But we realized that to get it into use, we had to do the next thing, which was to test on a smaller dose. I should say that there's a lot of evidence to suggest a small dose will work, but we needed a dose non-inferiority study to prove that. So we started that in 2014. And in fact, I saw Robin about 2 weeks ago, our last Newcastle recruit. We treated them for 2 years blind so we could look at the side effect profile accurately. And that meant a lot of very expensive aspirin packaging. And these tablets were either placebo or real so that we could give them different doses and they wouldn't know what dose they were on. Almost all of them agreed to stay on their dose for the next 3 years, which we had initially not intended to do, but the trial steering committee thought we'd get more power. And of course, we're using cancer as an endpoint, so it's perfectly reasonable for people to know what they're on. And indeed, most of them agreed to stay in. So we get the last patient, last visit in June from Melbourne, and then we'll break that down over the next few months to see whether it's true, as I suspect, that a small dose of aspirin will be effective. Why are old people different? Well, there are a number of reasons. And one of them is that our mitochondrial function breaks down in our colonic crypts as we get older. So that by my age, about 10% of my crypts will have defective mitochondria. Laura's been working on this for many years in our mitochondrial group, and we have a PhD student together looking at the impact of aspirin on an artificially old colon in a mouse, where we've got the polyp-forming mouse with a conditional knockout of mitochondrial function. Excuse me. What we already know is that if you knock out mitochondrial function, apoptosis goes down because it's depending on mitochondrial competence. And we also know that salicylates in nature are used by plants to drive programmed cell death to protect against infection. So losing apoptosis may well be important in the older colon. And crucially also, older people are much, much more likely to get side effects from aspirin. So we recommend not to use it beyond the age of 70. The other intervention we used was resistant starch or fermentable fiber. And this came from the work of Dennis Burkitt, the fiber man, who said that a high fiber diet of rural Africans protects them from colon cancer. And so, in fact, we used, both in CAP1 and CAP2, a so-called factorial design, like we used in the MRC vitamin trial in the 80s to prove that folic acid prevents spina bifida. And so a quarter of the recruits would take both interventions and a quarter of them would get neither, and then one for each of the other. And we didn't see any interaction between these two interventions, but we did see, surprisingly, an effect from the resistant starch as well. So these packets of powder were mixed with their diet for up to 4 years. And we had almost, 450 odd in each of the groups taking the starch. And we wrote a paper in 2012 saying we weren't seeing any effect around the world in the people who took the starch. But we had seen an effect in CAP1 on crypt cell proliferation. So we kept on going and we analyzed it in 2020. And to our surprise, it had worked as well, but not where we expected. It didn't have any effect on colorectal cancer, but it did dramatically reduce the non-colorectal cancers, which was our planned secondary endpoint. And so you can see there that, in fact, in this planned analysis, which was very important for the statisticians, we had a more than 60% reduction in other cancers associated with the Lynch syndrome. That particularly affected the upper GI tract, where there was a 75% reduction in upper GI cancers, which we think may reflect a change in the microbiome, causing a change in secondary bile acids, which are, of course, carcinogenic in this highly susceptible population. But whatever the mechanism, we did get a cover for cancer prevention research. Interestingly, the big journals were nervous of this because it seemed so improbable, but it really worked. And if the first half was good and got a prize, then the second half clearly was reliable as well. So we can prevent colon cancer with aspirin. We can prevent the other cancers with starch. Interestingly, we don't seem to make much of a difference with colonoscopy. So on the right here are the people who are in our prospective Lynch syndrome database, all of whom are getting colonoscopy. And on the left is a big study which looked at people who were not getting surveillance. These are very wide confidence intervals, but the crucial thing is there's not much difference in cancer risk between the left and the right, with the exception of PMS2, which is the least penetrant of the 4 genes. It seems like these patients get colon cancer almost regardless of colonoscopy. And interestingly, the more frequent you do scopes, the more cancers you find, suggesting that immunologically, a lot of these cancers may actually naturally disappear. And if you actually look back at our graph from CAP2, you see the same effect. Over 20 years, we saw almost no change in the placebo group in the cumulative incidence of colorectal cancer. So although we've now switched to more effective colonoscopy techniques, I suspect we won't prevent many cancers with colonoscopy, but we will, of course, detect them early and allow people to be treated before they die. So one of the questions is, why do 20% of these patients not get cancer when they've got problems all over their body? And our partners in Heidelberg did some beautiful work which showed that, in fact, about every square centimeter, excuse me, there is an aberrant crypt where you've got loss of mismatch repair function. So you've lost both copies of the gene, but you are still looking morphologically normal. And we think that these lesions can sort of spontaneously progress to cancer quite quickly without going through an obvious adenoma stage. And we put together all our resources to produce this paper from Aysel and Matthias, who are in the Heidelberg group. And what we came up with, or what they came up with, was that there are perhaps 3 ways to get to cancer. The obvious one, the usual one along the top, is you get an adenoma that then loses mismatch repair function and progresses to cancer, or indeed progresses to cancer without losing mismatch repair function. But then the bottom 2 are probably the more common ones, where you get a mismatch repair deficient crypt, which either briefly transitions through a sessile adenoma into a carcinoma, or indeed, as we see in mouse models, just progresses straight to carcinoma. So no matter how often you do colonoscopy, you won't prevent those. The immune response is becoming ever more interesting, especially as the vaccine story emerges. So the first thing that Matthias and colleagues showed was that one of the genes that contains a repeat is B2M. And losing B2M means you lose HLA expression, so you've got an invisibility cloak. So locally, the tumors can grow progressively. We also took colonic biopsies from our CAP2 patients and found that, weirdly, the ones that have more cells in their lining of their normal mucosa seem to be less likely to progress to cancer. It's not a very strong correlation, but you can see the graph at the bottom from our gastroenterology paper, which kind of makes sense. If you've got really good immune surveillance and you can see these cells emerging, you're more likely to kill them. And so that might explain why some people get cancer and others don't. The other reason for finding Lynch patients, of course, is that and I don't need to tell this audience about the revolution in immune checkpoint inhibitors. And so deregulating your T cells using Pembro or one of the other drugs that will knock out this protective mechanism means that these drugs can destroy microsatellite unstable cancers, whether they are sporadic or inherited. And indeed, a paper 2 years ago showed 13 patients with rectal MSI high cancers that didn't require surgery. They seem to be able to be treated medically. So finding Lynch patients has become even more exciting. I've been part of a group over many years, more recently led by Claire Turnbull at ICR, and also my colleagues who've been running the transformation program, to find these patients with Lynch syndrome and develop a more formal registry. They can all be tracked now by the NHS Disease Registration Service. And this is a fantastic piece of work done by Catherine and Lucy, which basically identified 9,030 gene carriers across England up to August of last year, which is a tiny proportion. Now, all of those are now going to go into the National Bowel Cancer Screening Program, so we can track them more easily. But in a population of 56 million, you'd expect a hell of a lot more Lynch patients. There should be at least 150,000 adults, even allowing for a shortened lifespan. So we're not picking these patients up. And that was what drove NICE in 2017, with a bit of prompting, to recommend that all colorectal tumors should be tested for mismatch repair deficiency. This is, by the way, just showing that these are some of the data around this Lynch registry, which I won't dwell on, except to say that there is an obvious patchwork quilt, as usual, across England. So in some centres, we're not picking up anything like as many Lynch patients as we should. The Southwest have got their molecular genetics well organised, are well ahead of the pack. And hopefully with what I'm about to show you, we'll be catching up soon. So how do we test tumours to see whether there is a mismatch repair deficiency? Well, Mark Aarons, of course, is an international authority on this. And thank you, Mark, for the slide, which is in the top left there, showing typical staining with the MSH2 antibody. The other technique that's been used for the last 20 years or more is the Promega fragment length analysis using MSI testing on 5 very large markers. And of course, you can also identify the signature of mismatch repair deficiency by whole genome sequencing, or indeed look for the genes themselves. All of these present us with a problem. Cell pathology is under great stress. I don't need to tell all of you, and only 3% of the labs in England are actually up to full strength. The Promega assay will not run through the new technologies. They're too big to put through a high throughput sequencer. And they also have this stutter peak problem, which means you have to have an interpretation by an expert. And ideally, you should look at normal tissue as well. And sequencing works, but it's expensive and you need a lot of DNA. So we set off about a decade ago to say, can we develop an MSI test that will run through our big machines? And we've succeeded in doing that. And the need becomes ever more obvious when you look at this paper we just published in the European Journal of Human Genetics based on the 2019 data in England. Of the 37,000 people who had colon cancer, we'd expect at least 32,000 of those to end up getting pathology examination. So you could do mismatch repair testing, and that should turn up at least 1,000 new patients with Lynch syndrome. What we actually found was that less than half of them actually got tested, and only 210 found their way to the end of the pathway and actually got a germline test done. So we're missing about 90% of the patients. We in the Northeast weren't doing that well. We were down at the bottom of the graph with only 30%, just under 30% being tested. Nick West and colleagues in Yorkshire got themselves really well organised with their IHC testing with Yorkshire Cancer Support. And as I say, a number of these never get to pathology because they're only imaged and then allowed to die. But nevertheless, it clearly wasn't working well. So our new test called the MSI Plus combines MSI testing with BRAF V600 and the driver RAS mutations. And these are my academic colleagues who drove this along. Mauro, in particular, a computational biologist, came up with 2 ideas which we patented. One of them was to use markers that were short enough to sequence and link to a polymorphism so we could differentiate noise from signal and get rid of that stutter. And on the back of that, we got a grant from the NHSE to roll this out nationally in England. And we've now got it installed in Northwest, Southwest, Central and South and North Thames. And hopefully it will spread to all of the GLHs. And we've got funding for a technologist and a minimum of 1,000 tests to be run. But we quite quickly found that the assay we developed using what's called molecular inversion probes was a bit clunky. So we developed a new one. And this is based on Rachel Phelps and Richard Gallen's work in our lab. And the idea, again, from Mauro was to sequence children with constitutional mismatch repair deficiency. These are children who get Lynch syndrome from both parents and get cancer from infancy. And they get an MSI high signal in their blood. And we used the markers that showed up in their blood repeatedly to pick the ones that we should test. So left to right here was our original 24 marker comparison between the blue stable and the red unstable. And you can see that there's not much of a gap, whereas north to south are our new markers, which are dramatically different and indeed completely eradicate the concept of MSI low, which many people refer to as really just an artifact of the old test. So instead of doing Promega and then reflex BRAF testing, which took a long time, and then separately doing KRAS, NRAS testing, we actually put it all together in one very simple one-day turnaround lab work. So we're getting results very quickly. And Lizzie, now in Northwest, then in Sheffield, designed this very nice lab screen for us. So basically, it compares the MSI result in this patient to all the others on this run. And we also see whether BRAF is present, as it is in this case, so we can exclude the need to progress to Lynch testing. And we get a QC. And you can see there the other 22 RAS driver mutations, which we're testing in the same test. We're also able to do this on biopsy blocks. So here are 4 Lynch syndrome biopsies where we individually test the biopsies. And as you will appreciate, sometimes you miss the tumor. But by taking curls from the whole block, we inevitably will get at least one of them showing an MSI high signal and a very clear signal. And this is mathematically calculated using Bayesian probability rather than just looking at it. So it's a yes/no test. And lowest there is now, I call Robotech. We've now repurposed the TCAN robot from our COVID testing lab so we can handle many thousands of samples with a technologist-led service. And in the northeast, North Cumbria, a less than 7-day turnaround. And this is what the report looks like. It says this one's got instability and it's BRAF negative, so increased likelihood of Lynch syndrome. And oh, by the way, don't give them an EGFR receptor blocker because they've got a RAS mutation. So this goes straight to MDTs and seems to be very well received. And about 5.5% of our cancers patients are now being referred for germline testing with almost complete coverage. So how am I doing for time? Not too badly. So what about other tumors? These are 4 genes which produce a really quite varied phenotype. And indeed, we probably shouldn't lump them together as one condition. PMS2 is a bit of a bridesmaid. And because there are 16 genes in this complex, you get redundancy, and particularly MSH6 can be replaced by probably MSH3, for example. So you get a less severe phenotype with MSH6 and PMS2, and we don't start colonoscoping them until they're 35. But it's more complex than that because there are a whole range of other tumors, not just colorectal and endometrial. And if you look at our prospective Lynch syndrome database, you can see that fairly dramatically illustrated. Just looking at the 3 big genes, I should say, and apologies to Ringo, I am a drummer in my spare time, and I know drummers are important, but he's clearly not as important as the other 3 in the Beatles. So cumulative risk for bowel cancer in blue here, you can see across all three of these genes. And endometrial cancer is showing here in orange, and again, in women, a very important risk. But look at the yellow ones, the urothelial cancers. 25% of men and 24% of women with an MSH2 mutation will get a urothelial cancer. And indeed, overall, 1 in 20 people with Lynch syndrome die of urothelial cancer. And up until now, we haven't even been able to do MSI testing on the tumors. So I think that's now going to change in the new test directory. So we said, since our test is now so sensitive, will it work as a urine liquid biopsy? And clearly that's important because the European urology community have issued guidance to say that all MSH2 gene carriers should be tested with ultrasound and dipping their urine for urinalysis and urine cytology. The people at the Mayo who see their patients every year have been looking at their urine, and they found that 9.3% of them had hematuria. None of those had urothelial cancer, whereas 5 of those patients over a couple of year period developed urothelial cancers, and they missed them all. So for those of you who are not into mathematics, that's 0% sensitivity and 0% specificity. So clearly not much use. It's probably a bit better than that, but clearly not good enough. So Alan here was one of our patients in the CAP study, and he got a kidney cancer. And so we asked him to send us urine. And you can see his pre-op urine there showing lots of the markers in our assay showing a high variant allele frequency. In other words, they were unstable. And that was matched by the markers from the tumor. So clearly we were seeing MSI from the tumor, which disappeared in both the urine and never appeared in the blood once he'd had his kidney out. And just looking at one of those markers, LR52, you can see the fairly obvious profile of the graph at the bottom, showing how the normal variant version of the allele has been destabilized and got multiple shorter versions, typically, of that particular repeat. And up to about 12 letters of the same type, you can put through a high throughput sequencer. So this is one of those short repeats. By coincidence, a young scientist who I'd seen about 10 years earlier contacted us to say that she'd been bleeding from her womb for about 6 months, and her GP said it was down to a fibroid. And we looked at her urine, and you can see in both Strec and Colli-P, these are shipping sample tubes, we could see, again, very obvious, very high numbers of variant alleles, which was also present on swab, but not on her mouth swab. And so she, in fact, had an emergency hysterectomy, and they identified a high-grade mismatch repair deficient cancer, which required radiotherapy, and clearly would probably have killed her, given that both the gynecologists she saw gave her reassuring advice. So we set up a pilot study in Freeman Hospital, where until last November I was the chair and continued to be a consultant. And we asked all our patients with MSH2 mutations to join in a routine clinic set up with Rakesh here, run by our clinical fellow, Rebecca. And with Bhavan and Arjun, we arranged to collect prospective urine samples from all of the people coming through with upper tract urothelial cancers to have a look at the urine. So these are the first 18 patients who came through with upper tract cancers, and you can see there that the pre-op urines in 2 of these were very high, as were the tumours themselves. The first was a 72-year-old who turned out to be an MSH6 Lynch syndrome patient and had a maternal history of endometrial cancer, which hadn't been picked up on. The other one was a lady who presented with both a bowel cancer and a urothelial cancer at the age of 56, and she turned out to be an MSH2 gene carrier, and there's her colorectal cancer and her ureteric cancer shown on the scan there. So basically, it clearly has value in terms of identifying Lynch families among the urothelial cancer referrals. And what about the asymptomatic Lynch patients? Well, we had 6 that out of the first 82 that we tested that were abnormal. One of those actually presented with frank hematuria and a bladder cancer just before he got to our clinic. But we did his urine test anyway, and he still had a high signal, which is test 1. They went back in and found he had bladder cancer that they'd missed. They couldn't get it all out, and so in fact, it was still high on the second sample and went back to the very high level on the third. So sadly, he is now in palliative care. This lady was strange. She came to the clinic and had a very slightly elevated assay in her urine, which actually went away spontaneously when we tested it a couple more times. And when she had her imaging, she turned out to have what looked like an exciting lesion in her kidney, which is a Bosniak cyst. But that had been there for at least 4 years. So we think maybe an incidentaloma. And it may be that she spontaneously destroyed a small urothelial cancer immunologically, as we were discussing earlier. All of the others had overt cancer. This 67-year-old will have his kidney removed next week. Sadly, he'd already had 1 kidney removed with urothelial cancer, so will now be going on dialysis. This lady had a 7.4 centimeter renal cell carcinoma, which is not the sort of cancer we'd expect to see. But removing it and the ureter took away the urine signal, although there was maybe 1 positive. And I heard yesterday that they found a cancer in her bladder, so it may be that she's got a quite separate urothelial cancer in her bladder. Whatever, we've basically done her a favor by picking her up asymptomatically with these cancers. This lady has 5 urothelial cancers in the renal pelvis on one side and is awaiting nephrectomy. Perhaps most exciting, this 47-year-old had a lesion in his ureter, which was successfully removed endoscopically, and his urine went negative. So we've picked up 4 asymptomatic urothelial cancers in this group, which is a little higher than expected, but nevertheless, of course, these cancers could have been around a while. And we picked up 2 Lynch families in the unselected upper tract tumours. We're now setting up to do a urine collection more widely and to also develop the assay to be more specific to the bladder and urothelial cancers by adding in some markers that will be useful there. And we put in a grant from UKRI to see if we can roll this out nationally. These samples will travel in the post for up to a week. So we're going to do this out of Newcastle, and you've got to have a good name. So we're calling it the MSI Urine Surveillance for Cancer and Lynch or musical study. And this is in part a tribute to our grandchildren, the wonderful Marsh family. This is them in the video where they made a video dedicated to my prostate cancer, which was successfully removed just over 5 years ago. And I would commend them to you. They are the most famous members of the family now. So these are all the people who made this possible. That's the people who developed the MSI Plus assay. And these are all the people involved in our CAP program. So in summary, Lynch syndrome, 4 genes, varying phenotypes, 600 milligrams will halve your risk. We'll tell you the best dose soon. Old colons lose their mitochondrial function, so may respond less. Resistant starch will cut your non-colorectal cancers, so keep eating the fiber. Metastasis is slow and rare because these frameshift peptides are highly immunogenic. T-cell infiltration may predict your cancer risk, and vaccines are under development. Immune checkpoint inhibitors clearly work. We need to find these patients. Scopes don't prevent cancers, but they pick them up early. And we've replaced immunohistochemistry completely in our region and saved about £600,000 for our 8 hospitals by switching to this much cheaper, simpler test. But obviously it can be used as in a complementary way to the immunohistochemistry, which is probably the best thing we can do if we can afford it. MSI Plus can identify urothelial cancers, and we're now piloting screening nationally, and hopefully many of you will be involved. And thank you for your attention.
Thank you very much indeed, John. That was a brilliant lecture and perfectly on time as well. Absolutely fantastic. If we were in a live setting, I'm sure you'd be hearing a very loud round of applause from all our audience members. So thank you very much. So I'd like to move on to the question and answer session next. I'd like to kick off, if I may, first with a question. You mentioned when you were looking at the starch that the starch is not effective for preventing the colorectal cancers. So I'd like to ask you what your thoughts are on that. Is it the microbiome or are other factors involved in that, do you think?
Yeah, thanks, Mark. And well, actually, that's what stopped us getting a second Lancet paper, of course, because the referees just couldn't believe that it wouldn't prevent bowel cancer, but could prevent the others. I think if I could take you back to that mechanistic slide, I think if you get colorectal cancers effectively emerging as cancers, then it may be that changing the microbiome will have less impact on that type of cancer, whereas the more traditional polyp, adenoma carcinoma progression might be more amenable to influence by the starch. I think the story that we came up with to perhaps account for the upper tract lesions was that changing the microbiome, which is clearly the only thing we could have done to have this very long-term effect, is perhaps making you less likely to produce secondary bile acids, which are reabsorbed and released into the bile, as you know. And of course, what these patients get are hepatobiliary cancers, duodenal cancers, to a lesser extent, pancreatic and gastric cancers, which would kind of fit with a carcinogen chronically being released in the upper GI tract. So it's a hypothesis. We don't know why, but the fact is it's cheap and effective. So we just tell everybody to eat a high fiber diet, basically.
Okay, great, thank you. So I'll move over to the Q&A box now. Our first question is from Matthew Brown. And Matthew says, is there any evidence for the use of aspirin and or starch for cancer prevention in the general population rather than those with cancer syndromes?
Yes, absolutely there is. I didn't have time to show you all of the other, well, actually I did show you the Gabriel Kuhn lecture, which of course was our presentation, which said that people who took a lot of aspirin get less bowel cancer generally. There was a second trial with cancer as an endpoint set up in America called the Women's Health Study, and it didn't show an effect of baby aspirins over 10 years, but in the second decade there was a 20% reduction in bowel cancer in the people randomized to small aspirin. And then the other crucial data came from Peter Rothwell, who went back on a multitude of old cardiovascular trials and tracked down no fewer than 25,000 people who'd taken part in aspirin trials in the 80s and early 90s. And in that group, there was a significant 20% reduction in colorectal cancer death in the following decades. So we've got clear evidence this is a generic effect. The problem is the side effect profile. But in your 50s, basically, Jack Cusick led a paper we published in 2015. If you gave everyone between 50 and 60 a baby aspirin a day, you would cut the national mortality by 4%. The effect on aspirin is actually bigger than the effect on cardiovascular risk. But it's always that slight anxiety about GI bleeds, particularly in the elderly, and the very slight anxiety of subarachnoid hemorrhage, which is largely confined to the hypertensive population.
Thank you. Okay, so our next question is from Maurice Lockery, who says, thanks for a wonderfully informative talk. Any ideas why some LS-related colorectal cancers harbour RAS mutations, but are supposedly never found to harbour a BRAF mutation? Are we missing any Lynch cases in following the NICE pathway, not testing those with BRAF mutations?
Yes, well, you're absolutely right. And we are missing some because clearly you can get a RAS mutation or a BRAF mutation in any cancer. You know, once it becomes a sort of chaotic situation, anything can happen. The point is that from a mechanistic point of view, you're not going to get that type of mutation typically in a mismatch repair. It's mostly the repetitive DNA that breaks down. So yes, we will miss a few. On the other hand, we avoid a deluge of referrals for germline testing. We simply can't cope with germline testing everybody. But it is an argument in favor of immunohistochemistry. Conversely, immunohistochemistry doesn't actually test function, it tests the protein's existence. And so there is also a documented miss rate with immunohistochemistry. And in Ohio, where we've done a lot of work together, they actually can afford to do immunohistochemistry, MSI testing, and sequencing. And so what they've demonstrated is that MSI and IHC together will pick up everything virtually. If you'll miss 2 or 3% with either of those tests used in isolation.
Thank you. That's a good, clear answer. Thank you. So next question is from Olivier Geiger, who says, how many of the patients who received MSI plus testing still get NGS testing after an MDT discussion?
5.5%. Well, 5.5% are recommended to go forward for germline testing. And crucially, what we found in our 2019 audit was that it was like months before we got to the germline test question. We're now mainstreaming with our nurse practitioners. So basically they get told this person should be offered Lynch testing and they're still under the care of the hospital. In fact, in many cases haven't yet had their surgery. And so it's going to be a much higher pickup rate. Not everyone will agree to be germline tested, but my experience is while someone's being treated for cancer, offering them a germline test, which could influence your choice of surgery or indeed whether you do surgery, is much more likely to be accepted. Whereas if you approach someone 2 months later when they're in recovery and back at home and need to come to the hospital for a blood test, etc., it just doesn't happen. So we can't give you a definitive answer yet, but almost certainly this much faster turnaround and yes/no advice will make a big difference.
Thank you. Okay, so next question is from Neil Shepherd. And Neil says, John, great talk. Is your Lynch screening test to be rolled out nationally? We do 2,000, i.e. 500 patients mismatch repair immunostains a year, and they're very expensive. So can we use your test being rolled out nationally?
Yes, please. And Mark, as you know, we've got a presentation in Scotland and we've been talking to the Welsh as well. As you all know, trying to change pathways is a nightmare in an overstretched service. But if anyone outside of the Northeast, North Cumbria or Yorkshire, where we're already in conversation directly, and we will provide the MSI testing in the Northeast, Yorkshire Northeast GLH. But we've got it set up now in Northwest, it's set up in Bristol, it's set up in, I think it's set up in the Marsden now, and it's set up more or less in Birmingham. So I'd really like to hear from you if you'd like to do it in your hospital and are struggling at all to get it rolling. It always takes a while to get these things moving. But if you were to use it, I mean, the point is it's funded by the Genomics Service. It's on the test directory. You can have the test and you get all, and crucially, Nick West, who will be known to all of you, who did that wonderful work in Yorkshire, is switching completely to MSI instead of IHC. And as I said, it costs less than half to do this test. It's not much more than the cost of the antibodies to do the whole test for the MSI plus. So it's a big saving for your hospital because, of course, it'll be outsourced to the GLH, but also it's a big saving for the NHS because it's a half the price test.
That's a great answer. Thank you, John. And you mentioned Nick West, and so this is a good segue into a question from Nick. He says, fantastic talk. Thanks, John. We are strong supporters of the MSI plus assay in colorectal cancer, having tested 800 plus of our cases to date. Can you clarify for other regions of the UK outside northeastern Yorkshire how pathologists can ensure that their GLH/genetics service is set up to deliver this it is not as widespread as it should be, in my view, says Nick.
Thank you, Nick. And yes, I agree. And that's why I said before, it's getting these things going is hard. We have funded from our grants Birmingham, Manchester, Marsden and Bristol. Bristol have now gone live. Just a technical issue, we set this up on the MySeq, which will run, and that's why we wanted people to do it centrally, because you can then do about 40 at a time. But we're now adapting it to the next Seq, which is the bigger machine, and it means you can chuck our markers in with everything else. So it doesn't matter whether you've got 10, 12, 40 or whatever. And so almost certainly the other labs will use the next Seq approach. We'll put the software in an NHS cloud so they can access it. And make this into a commercially viable test in the NHS. People will basically pay us every time they issue a report, and the hospital will get, a few quid. And that will then allow us to package this to sell it into other jurisdictions in the States and Europe, etc. So essentially, it's good to go. It should have gone. It's gone in to some extent, but I'd love to hear from pathologists, because I think it needs a bit of pull from the leading pathology community, rather than saying, we're fine, we do IHC, go away. Because as you said, it is expensive IHC, and it's a demanding thing to add to the list of things you guys have to look down a microscope for. So anything that takes weight off your shoulders seems to me like a good idea.
I couldn't agree more. I think that's a very good answer. Thank you. So moving on to the next question from Lach Ilyas. Lach asks, do LS bladder cancers have the same relationship with BRAF mutation as in colorectal cancer?
No, they don't. Thank you. And that's what I mean. Basically, the BRAF driver is very much a colorectal selector. And looking at RAS mutations is actually quite a useful thing to have, obviously, from an oncology point of view, because RAS and MYC are two of the sort of common pathways that in all cancers. But no, that's one of our problems with the bladder. Our other markers are designed to pick up colorectal. I didn't mention endometrial because NICE said you have to use immunohistochemistry in endometrial. That's partly based on old data. Our assay is actually slightly better in terms of accuracy than immunohistochemistry in endometrial, but crucially, we don't have any other markers to work out which one should go for germline testing, except methylation testing. So we can't use it in endometrial at the moment because we're not allowed, well, we can, but we are using it locally and in Edinburgh. But that's sort of research work. Everywhere else, for the urothelials, yes, we're dependent on MSI only, and that's why we want to add new markers like TERT and so on, which will allow us to be more selective and indeed pick up bladder cancers that are not MSI high, because of course these patients will get bladder cancers anyway and will miss a bunch if we just rely on microsatellite instability. And indeed also it means the assay could be used more widely in a wider population. And indeed, I'd quite like to see it being used in women with dysfunctional menstrual bleeding in their late 30s, because clearly a bunch of them will have an MSI high. About 1 in 3 endometrial cancers are MSI high. So you're going to pick up a bunch of sporadic endometrial cancers in that community as well.
Would you consider working up an MLH1 promoter hypermethylation test as part of a next generation of MSI plus?
Yeah, absolutely. Well, so the problem with, as you know, Mark, to do methylation testing, you've got to use a second digest. So it becomes a second test. But absolutely, one of the things that we would do if we brought it in for endometrial cancer is simply do the endometrial MSI and then do the methylation test on the DNA we've got in the lab. And one of the reasons we got the grant from NHS England was the point was, you've got to give pathology departments a reason to send samples to the molecular lab. And here is a good reason to send it. Once it's there, then you can do other stuff on that molecular sample. So trying to build in routine sectioning of every solid tumor to do 3 curls to molecular without thinking about it means that you can actually widen your circle of testing. And it only costs about 10 quid to do the curl cuts and 10 quid to do the extraction. So it's a relatively cheap thing to add in as a standard of care, I would dare to suggest, for all of our solid tumours.
Yeah, thank you. Okay, so I've got a comeback from Olivier Geiger, who says, sorry, I was referring to somatic tumour testing. So if he could revisit his earlier question. His earlier question was, How many of the patients who received MSI plus testing still get NGS testing after the MDT discussion? And I think he means NGS testing of the somatic tumours.
Well, in our region, none, as far as I know. But I wouldn't necessarily know whether they have done that, because that would be an oncology request direct to Leeds who do it. So Nick West and colleagues might be better able to answer that question. But I mean, basically, from the point of view of deciding who to do germline testing on, you don't need to do further sequencing of the tumor. As far as the colorectals are concerned, you've got most of what you need. But I'm perhaps not the right person to answer that question. I'm not averse to doing tumor sequencing. Remember, we're sequencing the biopsies, not the tumor. And so we would test the biopsies and say, this person needs germline testing. What happens to the tumor once it's resected is somebody else's story. And all I can say is that, everybody knows the genomic labs are up against it in terms of just the numbers they can process at scale.
Okay, thank you very much indeed, John. It's a few minutes to 10, so we'll stop there, and I'll just finish by thanking you again for an absolutely fantastic lecture, very informative, huge amount of data covered, really interesting, so thank you very much indeed.
My pleasure. Thanks for having me.
Thanks everybody.